Biological oxidation is a process which leads to formation of ATP in the body. So let us see how that exactly happens. So we have the electron transport chain which is located in the mitochondria. Okay. So if you look at the structure of mitochondria, you have two membranes. One is the outer membrane, the other is the inner membrane. And the space in between is known as the intermembranous space. Okay, so this is the intermembranous space. Now the electron transport chain is located on the inner membrane and it comprises of complex 1, 2, 3, 4 and 5. Now what actually happens in this complex is we have transport of electrons. Okay, so two electrons are transported through complexes. Okay, through complex 1 or complex 2, it goes through complex 3 and ultimately the two electrons it reaches complex 4 where it combines with oxygen to form as water, and that is why oxygen is essential for the electron transport chain to work. Okay. Now how does this transport takes place and what donates these two electrons? Okay, so let us see. This NADH and FADH2, they are the ones which donate the electrons. Okay, hence they are called reducing equivalents. So in our body there are various biochemical reactions which takes place and we have these reducing equivalents which are produced, NADH and FADH2. So these NADH and FADH2, they come in the mitochondria, in the matrix of the mitochondria, and then they donate the two electrons to the complexes, and then the electron transport chain process takes place. So let us first try and understand how NADH works. So the NADH, it donates two electrons to the complex 1. Okay, so NADH is converted to NAD plus 2 hydrogen ions, and in the process it donates two electrons to the complex 1. Now through the complex 1 you have the oxidation and reduction reactions takes place, and ultimately the energy which is released, that energy drives 4 hydrogen ions from the matrix to the intermembranous space. Okay, so these 4 hydrogen ions, they move from the mitochondrial matrix here to the intermembranous space. Then these two electrons are carried by the coenzyme Q, and then these coenzyme Q again carry the electrons from complex 1 to complex 3. Now on complex 3 also we have oxidation reduction reactions which take place, energy is released, and that energy is used to drive the protons, 4 H positive ions or protons from the matrix into the intermembranous space. The electrons are then picked up by cytochrome C and they are taken to complex 4. Again we have a set of oxidation and reduction reactions which takes place that we will see later, and we have is production of energy which drives 2 hydrogen ions or 2 protons from the matrix to the intermembranous space. So there are total production of or movement of 10 protons which occurs from the matrix into the intermembranous space. So when NADH donates 2 electrons, then there is movement of 10 protons from the matrix to the intermembranous space. Okay, so as a result, there occurs increased concentration of protons inside the intermembranous space as compared to the matrix. Okay, now let us see how FADH2 works. Now when FADH2 reducing equivalent, it donates 2 electrons to complex 2 and not complex 1. Okay, and again here the oxidation reduction reactions take place, but the amount of energy which is released here is not sufficient to drive protons from matrix to the intermembranous space. Okay, so these 2 electrons are picked up by coenzyme Q, the transport mechanism, and they are taken to complex 3. Okay. Here again oxidation reduction reactions take place, and we have is movement of H positive ions from the matrix into the intermembranous space. The electrons are further picked up by cytochrome C, taken to complex 4, where again you have is movement of H positive ions from the matrix into the intermembranous space. So when FADH2 is the reducing equivalent, then there are production of 6 H positive ions inside the intermembranous space, or movement of 6 protons from the matrix into the intermembranous space. Okay. After this, what happens is this results in increased concentration of hydrogen ions or protons in the intermembranous space as compared to the matrix. Now these increased concentration of protons, they come back inside the matrix through the complex number 5, which is also known as ATP synthase complex. And when they do that, this results in conversion of ADP to ATP, and this results in the production of ATP. And this is the reason why the electron transport chain, the function of it results in the formation of ATP, and mitochondria is known as the powerhouse of the cell. So biological oxidation is defined as transport of electrons from the reducing equivalents, that is NADH and FADH2, through the electron transport chain, which ultimately combines with oxygen, results in formation of water, and the energy which is released is used to form the ATP. Okay. Now let us see how the complexes they act. Okay, so again, these are the 4 complexes, complex 1, 2, 3, and 4. Now when the reducing equivalent is NADH, okay, so NADH is converted to NAD, and this results in donation of electrons. So when electrons are donated, see, it is NADH. The first complex is made up of flavoproteins and iron sulfur proteins. Okay, so the flavoproteins FMN is reduced to FMNH2. Okay, so this FMNH2 again undergoes oxidation, loss of 2 hydrogen ions. So we have is oxidized iron sulfur proteins, which take up these hydrogen ions and they become reduced. Now this reduced iron sulfur proteins again become oxidized. Okay, and the 2 electrons are ultimately picked up by coenzyme Q, and coenzyme Q becomes reduced. So in the complex 1, we have a series of oxidation and reduction reactions, okay, which results in formation of energy, which drives the 4 protons from matrix into the intermembranous space, as we have seen earlier. Okay, so this coenzyme Q now goes to complex 3. Okay, complex 3 coenzyme Q, the reduced form is again converted to oxidized form. It donates the hydrogen ions. So complex 3 again is made up of iron sulfur proteins. It is made up of cytochrome B and cytochrome C1. Okay, so this iron sulfur protein again, they change between oxidized state and a reduced state. Okay, so here this is the oxidized state. They accept the electrons, and they become the reduced state. Again, cytochrome B and cytochrome C1 also, they change from oxidized to reduced state, and ultimately the cytochrome C picks up the electron and it transports it. So the cytochrome C then transports the electron to complex number 4. So when the electron is transported through complex number 3, again you have this movement of 4 hydrogen ions from the matrix into the intermembranous space. Now the cytochrome C donates into the 4th complex, which is made up of again copper and heme. Okay, it is AA3 complex, which is oxidized and reduced, just like the earlier complexes. And this also results in release of energy, and that energy drives 2 hydrogen ions from the matrix into the intermembranous space. Okay, so this results in the movement of 10 protons from matrix into the intermembranous space. So when NADH is the reducing equivalent, there is movement of 10 protons from matrix into the intermembranous space. Okay, and this occurs because of the repeated sequences of oxidation and reduction which takes place in complex number 1, complex number 3 and complex number 4. Okay. Now when we have is FADH2 as the reducing equivalent. Now FADH2 donates 2 electrons to complex number 2. So here again the FADH2 and we have his iron sulfur proteins which become reduced and oxidized. Okay, so we have his oxidized iron sulfur protein which takes up the electrons, becomes reduced, and this, when it donates the electrons, it becomes oxidized, and that donated electron is picked up by the coenzyme Q and forms his reduced form of coenzyme Q. So from here in this complex 2, this oxidation and reduction reaction, the energy which is produced is not sufficient to drive the protons from matrix into the intermembranous space. So from complex 2, now the coenzyme Q goes to complex 3. Okay, so this is the coenzyme Q. So it goes to complex 3. Again, we have the same oxidation reduction reactions, and there occurs movement of 4 H positive ions across complex 3 from matrix into the intermembranous space. And the electrons are then carried again by cytochrome C to complex number 4, and you have this movement of 2 H positive ions. So when FADH2 is the reducing equivalent, we have movement of 6 protons from matrix into the intermembranous space. Okay, and like we have seen earlier, these protons which are now in increased concentration in the intermembranous space, it comes back to the matrix via the complex number 5. Okay, so how ATP is produced, that is given by chemiosmotic hypothesis that we will see next. So in the chemiosmotic hypothesis, okay, again this is a different figure of the electron transport chain. Here is the NADH or reducing equivalent, and when the electron flows through complex 1, complex 3 and complex 4, you have is movement of H positive ions in the intermembranous space. Okay, and we have is 10 H positive ions or protons which are present in the intermembranous space, and it comes back through the complex 5. Now this complex 5 consists of two parts. Okay, one is the FO domain and the other is F1 domain. So the FO domain is present on the inner mitochondrial membrane, whereas the F1 domain, this is present in the mitochondrial matrix. Okay, so let us look at the structure of FO and F1 domain. Okay, so the protons are coming back into the matrix through the FO domain. Now if you look at the structure of FO domain, so this is the FO domain and this is the F1 domain. So FO domain contains the C rings, okay, and the hydrogen ions pass through these C rings and it causes rotation of these C rings. Okay, the F1 domain, it is made up of various polypeptides, right from alpha, beta, gamma, delta, epsilon. Okay, so out of this, the beta polypeptide is of importance, to which ADP is attached. Okay, so what happens when the hydrogen ions, they move through the concentration rings, C rings? Okay, so the hydrogen ions, they cause rotation of these C rings. So when rotation of these C rings occur, this results in a conformational change of the beta polypeptide of the F1 unit, which results in conversion of ADP to ATP and formation of ATP takes place. So this complex 5 or ATP synthase, this is how ATP is formed. Okay. Now let us talk about PO ratio. What is PO ratio? PO ratio is phosphorylation oxygen ratio. So phosphorylation is the amount of inorganic phosphate or ATP which is formed per oxygen atom which is consumed. Okay, so we know that for formation of 1 ATP, what is already known is that 4 hydrogen ions are required. Okay, so the hydrogen ions, they move through this FOF1 complex and results in formation of ATP. So 4 hydrogen ions are required for formation of 1 ATP. Now we have seen that NADH produces 10 hydrogen ions. So the number of ATP will be formed is 10 divided by 4, that is 2.5 ATPs. So that is why 1 NADH results in production of 2.5 ATPs. Similarly, 1 FADH2 produces 6 hydrogen ions in the intermembranous space. Okay, so the amount of ATP which is produced is 6 divided by 4, that is 1.5 ATPs. So 1 FADH2 provides or gives 1.5 ATPs. Okay. So let us now look at the inhibitors of electron transport chain. Now this is the same electron transport chain. So we have inhibitors which affect each complexes. Okay, the first one is the complex 1. So the movement of electrons between the complex 1 and coenzyme Q is affected by these drugs. Okay, alkyl guanides, barbiturates, chlorpromazine, and antibiotic pyricidine. Okay, so if you can remember this as complex number 1 starts with A, so A, B, and C. A for alkyl guanides, B for barbiturates, C for chlorpromazine. So this inhibits the electron transport chain between the complex 1 and coenzyme Q. It inhibits the transport of electrons between complex 1 and coenzyme Q. Similarly, the movement of electrons between complex 2 and coenzyme Q is inhibited by carboxyn. Okay, and the movement of electrons from complex 3 to cytochrome C is inhibited by BAL or British anti-leucide. Okay. Now we have inhibitors of complex 4. So cyanide, okay, carbon monoxide, and hydrogen sulfide, they inhibit the complex 4 of the electron transport chain. So these are the inhibitors of the various complexes of the electron transport chain, right from complex 1 to complex 4. Then we have is inhibitors of oxidative phosphorylation. Now what is inhibitors of oxidative phosphorylation? These are proton gradient inhibitors. That means, so we have seen that when the electron transport chain process takes place, there occurs increased concentration of H positive ions in the intermembranous space. So we have increased concentration of H positive ions here as compared to the matrix. So a proton gradient has been formed, okay, according to which there is more hydrogen ions here in the intermembranous space as compared to the matrix. Okay, so then as a result of which the protons, they move from the intermembranous space through the ATP synthase complex back into the matrix, and this results in the formation of ATP as we have seen. So inhibitors of oxidative phosphorylation are those inhibitors which inhibit this proton gradient. Okay, so the first one is oligomycin. So oligomycin affects the FO portion of the ATP synthase complex. Okay, so ATP synthase or complex 5 consists of FO and F1. So the function of FO was to allow the H positive ions to pass through. Okay, so O means oligomycin. Okay, the O stands for oligomycin because that portion, that FO, is inhibited by oligomycin. So oligomycin inhibits FO, so the protons cannot move inside. Okay. Next is atractyloside. Now atractyloside, it inhibits the ADP movement inside the matrix. Okay, so this ADP actually is transported from the cytoplasm, it comes into the matrix, and then this ADP is converted to ATP by chemiosmotic hypothesis, that as we have seen. So atractyloside, what it does is it inhibits the movement of this ADP from the cytoplasm to the matrix. Okay, so as a result ATP formation is inhibited. Okay. Next is valinomycin. Now what valinomycin drug does is it is an ionophore. Okay, so basically it creates channel for movement of ions. Okay, so valinomycin creates a channel which allows movement of potassium ions inside the matrix. Okay. Now we have, before this we have a gradient, right? A proton gradient. That means more H positive ions or more positive charge in the intermembranous space as compared to the matrix. Now, if we have is melanomycin, what it does is it creates a channel which moves potassium ions into the matrix. So when this happens, the proton gradient which was created, so that gradient again is dissipated. Okay, so that gradient is lost because potassium also has positive charge. So the positive charge of the hydrogen which was supposed to come back like this, instead of that we get positive charge from potassium ions. Okay, so valinomycin is a drug which creates a channel. This channel results in movement of potassium ions inside the matrix. And when the potassium ions move inside the matrix, then the proton gradient which was created, that is lost. Okay. Next we have is set of uncouplers. Now what are uncouplers? Uncouplers are, okay, same thing. Now normally what happens is the H positive ions which are accumulating here, they go through the ATP synthase complex and we get formation of ATP. That is normal process. Now if instead of the H positive ions going through this ATP synthase, they go through a channel which is different channel. Okay, so when this happens, these are known as uncouplers. So uncouplers, they basically create a channel through which the H positive ions, they move from the intermembranous space into the matrix. But since they are not going through the ATP synthase, so ATP is not getting formed. Okay, but the gradient which was there, that gradient is lost. So the proton gradient which was created, that proton gradient is now lost because uncouplers, they provide a channel which allow the movement of H positive ions from the intermembranous space into the matrix. And when this happens, you have this increased production of heat. Okay, so energy which is released in the form of heat. So we have pathological uncouplers and we have this physiological uncouplers. So 2,4-DNP and 2,4-DNC are pathological uncouplers, whereas thyroxine, the thyroid hormone, that is physiological uncouplers. Okay, so that is why in case of hyperthyroidism, we feel more heat is there in the body. Okay, heat intolerance takes place. Why? Because thyroxine acts as a physiological uncoupler. Okay, and this allows the H positive ions to move inside the matrix, and the energy which is released in the form of heat. Similarly, thermogenin also is a physiological uncoupler. Now, thermogenin is present in brown fat, which is seen in infants. Okay, so these infants, they have brown fat. So this brown fat contains thermogenin, which is an uncoupler. So when this process takes place, there is again increased production of heat. So this is how infants, they keep themselves warm. So this is a protective mechanism for infants so that they can keep themselves warm. Okay, because they have increased brown fat, and brown fat contains thermogenin. Thermogenin is a physiological uncoupler. So uncouplers, they allow the H positive ions to come inside the matrix, okay, through different channel, and the energy which is released in the form of heat. Okay, so this was all about the inhibitors of the electron transport chain. Okay, I hope you have understood. So thank you very much, and I will see you soon.